Hello, everybody. Thank you for tuning into our presentation. My name is Spencer,
My name is Anna.
and we're going to talk to you guys about leptin receptor expressing neurons in the ventromedial nucleus of the hypothalamus and how they contribute to weight loss caused by fourth ventricle leptin infusions. Some good information to note in this experiment is that purebred male Sprague-Dawley rats were used as model organisms to better understand human hunger modulation. So how the research team did this is they used a plant toxin known as saporin, which is a ribosome inactivating protein that removes an essential adenine base within the eukaryotic ribosome, which irreversibly inhibits protein synthesis in the affected cells, causing cellular apoptosis. To target the leptin receptors, the research team conjugated leptin to saporin to act as a targeting agent. Once the leptin binds to its membrane or OBR receptor, saporin is then internalized through endocytosis, broken off from the leptin, and then inactivates ribosomes, causing apoptosis. Thus, OBR expressing neurons are targeted with this site-specific injection. Cells outside of the injection site without leptin receptors are not affected and survive this treatment. Blank saporin and PBS-infused rat groups represent negative controls to assess the impact of the experimental procedure alone on the rats. In normal physiology, leptin is secreted by adipose cells to reduce hunger drive and induce weight loss through the JAK-STAT pathway. The center for neurological control is the ventromedial hypothalamus, which is bathed in fourth ventricle fluid, as shown in the bottom right figure. Leptin receptor ablation results in hyperphagia and weight gain, as is seen in the top right-hand rats. This is the JAK-STAT leptin signaling pathway. First, leptin binds to its receptor, which dimerizes and then phosphorylates a STAT protein, which itself dimerizes and acts as a transcription factor to promote downstream gene expression. The researchers' objective for this experiment was to determine the importance of the ventromedial hypothalamus in the central response to leptin, and specifically in the weight loss caused by fourth ventricle infusions. The researchers' hypothesis was that weight loss in leptin-treated animals only occurs when there is simultaneous activation of receptors in the hindbrain and forebrain.
So now we will be discussing what researchers did prior to the experiment in order to set up the proper parameters. The appropriate coordinates for injection and the appropriate injection volume of saporin were first found.
Leptin saporin diffusion and localization was assayed using immunohistochemistry via fluorescing antibody specific to the OBR receptor, which is the leptin receptor. Following this, in situ hybridization showed regions of lack of OGR expression. This showed the team that leptin saporin injection did not deplete leptin receptors outside of the ARC or the DMH regions of the brain. Calorimeter cages showed rat energy consumption within each treatment group, and this data was monitored throughout the course of the experiment.
Next, the researchers prepared the rats with bilateral VMH injections of leptin saporin or blank saporin and recorded the rats' daily weights. Infusion cannulas were then fitted to the rats and a glucose tolerance test was performed. The rats' food intake, energy expenditure, and respiratory exchange ratio were then recorded. On day 0 of the intended experiment, pumps injected either isotonic saline or daily leptin. Food restriction was then enforced, and fat sections and brains were collected and assayed. Statistical analysis was then performed. On this slide is an overview of the statistically significant results obtained by this experiment. It was first found that there was no statistical significant effect of VMH leptin saporin on weight gain or glucose clearance before leptin infusion. It was found that upon infusion of leptin, food intake and respiratory exchange was inhibited in blank saporin rats, but not in leptin saporin rats. Leptin was found to have no effect on the energy expenditure or brown adipose tissue temperature in either of the groups. Additionally, it was observed that inguinal and epididymal fat levels were significantly reduced in leptin-treated blank saporin rats, but the response was reduced in leptin saporin rats. Finally, VMH p-STAT3 levels were observed to increase leptin-treated blank saporin rats, but not in leptin saporin rats. Now, we will discuss figures that the researchers used to visualize the obtained data. The researchers performed a glucose tolerance test in order to observe baseline metabolism rates of the rats before the intended experiment began. The rats first were injected with glucose and monitored in 15-minute time intervals for 120 minutes. It was found that in the blank saporin injected rats and in the leptin saporin rats, there was no significant difference in basal metabolism rates. This allowed researchers to perform the experiment knowing that metabolism would not affect the weight fluctuation of the rats, rather, that leptin was the primary influencer of weight loss. In this figure, the daily body weights of the rats in each treatment group are recorded. Here, we can observe that the rats maintained a steady body weight before infusion of leptin or PBS. The blank saporin rats and the leptin saporin rats that were injected with saline did not lose weight overall, as expected. Rather, the PBS-infused blank saporin rats stopped gaining weight during the first 5 days of infusion, but then started to gain weight at a steady rate. Leptin saporin rats receiving PBS infusions gained weight during the second week of infusion and weighed the same as the blank saporin PBS-infused rats at the end of the experiment. Leptin-infused blank saporin rats lost weight throughout the experimental period and weighed significantly less than any other group during the last 5 days of the experiment. Leptin infusions in leptin saporin rats inhibited weight gain but did not cause weight loss as experienced by the blank saporin leptin-infused rats.
Here, we see visual representation of the effects of conjugated saporin on leptin receptors. On the left side, we see many more black dots in the ventromedial hypothalamus, which represents increased leptin receptor density. This is expected since the blank peptide sequence should not target leptin receptors. In the right figure, brain sections had leptin conjugated to saporin dose, which ablated those neurons, producing no observed black spots. Results of this experiment support the hypothesis. Leptin-treated animal weight loss occurs when there is simultaneous activation of both hindbrain and forebrain receptors, suggesting that the response is controlled by multiple brain areas. Results support previous findings that ventromedial hypothalamus leptin receptors have a small, if present, role at maintaining hunger and energy balance in normal homeostatic conditions. Findings support that VMH leptin receptors limit weight gain during times of hyperphagia.
Now, we will discuss concerns with the experiment that may warrant further research. The researchers only tested male Sprague-Dawley rats, and leptin is known to express differently between the sexes. In the future, female rats may need to be tested in order to view the full impact of this experiment. Additionally, in the initial tests performed to set the experiment's parameters, statistical analysis was not performed. Finally, some of the rats had to be removed from the experiment because of food intake issues, possibly due to a misplaced cannula. Now, we will pose our discussion questions. First, what is the purpose of the glucose tolerance test to understand metabolism in the tested rat population? Briefly describe this test. What are the human applications of this test?
Second, why do you believe fat levels were significantly reduced in leptin-treated blank saporin rats? Why was VMH pStat3 concentration increased in leptin-treated blank saporin rats, but not in leptin saporin rats? Thank you much for your attention. We hope you enjoyed our presentation.